Deep inside every living cell lies a tightly coiled, incredibly complex instruction manual, DNA. For decades, biologists could only stare at this manual. They could observe the code and record its outputs, but they completely lacked the tools to extract or copy specific sequences. This limitation created a severe medical bottleneck by the mid-20th century, particularly for the millions of diabetics who depended on daily insulin. Pharmacies didn't have synthetic options. The only way to get insulin was to harvest it directly from the pancreases of slaughtered cows and pigs. It was a brutal, wildly inefficient process. Extracting a tiny vial of medicine required mountains of animal organs. Because the resulting insulin came from livestock, patients frequently suffered severe allergic reactions. The irony was that the human body already held the pristine code to produce pure insulin. We just had absolutely no way to snip out that specific instruction and manufacture it. Medicine had hit a hard biological wall. To move forward, we needed a way to literally copy and paste nature's own blueprints. In 1973, Stanley Cohen and Herbert Boyer figured out a workaround to this biological limit. They developed recombinant DNA technology. In plain terms, taking DNA from different organisms and stitching them together into a single molecule. Instead of trying to synthesize complex chemicals from scratch in a lab, Cohen and Boyer realized they could trick bacteria into doing the heavy lifting, turning them into automated, microscopic production lines. Rather than a static structure viewed under a microscope, the cell became a programmable manufacturing plant. The process starts with isolation. Using a technique called PCR, scientists scan a massive donor genome, identify the exact target gene, like the sequence for human insulin, and amplify it, essentially printing thousands of copies of that single instruction.
But that raw genetic code needs a biological taxi to transport it into our factory. For this, we use a vector, typically a circular ring of bacterial DNA known as a plasmid. To combine them, scientists introduce restriction enzymes. These act as highly specific molecular scissors. They cut open the circular plasmid and snip the ends of our human target gene, creating jagged, matching overhangs called sticky ends. In the third step, ligation, an enzyme called DNA ligase acts as biological glue. It stitches the human insulin gene perfectly into the gap in the bacterial plasmid, sealing the loop to create a brand new recombinant plasmid. We now hold the modified instructions in our hands. The next step is getting a living engine to actually read them. That engine is usually E. coli. It's a common bacteria, but it serves as an incredibly efficient host for our new instructions. To get the plasmid inside, scientists use a process called transformation. They take the E. coli and rapidly hit them with a blast of heat or a tiny electric shock. This forces the bacterial cell walls to briefly open their pores, allowing the recombinant plasmid to slip inside. It's an imperfect science, though. Out of millions of bacteria in a sample, only a tiny fraction will actually absorb the plasmid. To filter out the failures, we use selection. By growing the bacteria on petri dishes laced with antibiotics, scientists use a method called blue-white screening to kill off the empty cells and isolate only the successful modified clones. Once those successful clones are isolated, biology takes over. Every time that bacterium divides, it replicates our newly inserted human gene, scaling up a microscopic edit into exponential mass production. This diagram outlines the genetic flow mapped out in 1978. Following this exact assembly line, scientists successfully guided an engineered strain of E. coli to synthesize active human insulin for the very first time. Today, those small petri dishes have scaled up into enormous stainless steel fermentation tanks. Millions of gallons of engineered bacteria are cultivated simultaneously. This industrial scale completely eliminated the organ harvesting bottleneck. It guaranteed a consistent, safe, and virtually unlimited supply of human-identical medicine for diabetics worldwide. The insulin success proved the method worked. Gene cloning quickly became the base mechanic for the entire modern biotech industry, enabling scientists to produce disease-resistant crops and targeted gene therapies. By learning how to cut, paste, and run biology's native code, we stopped merely observing the blueprint of life and finally began engineering it.